Researchers look at humans and see us as special or different, and therefore slightly different rules might apply. And as part of my general model that humans are animals, we should also be treating the taxonomy of humans the same way.
Hello, today I have the pleasure of speaking with Dr. Brian Villmoare of the Department of Anthropology at the University of Nevada, Las Vegas. So welcome, Dr. Villmoare. Could you please share a brief overview of your career and research?
Sure. I'm a paleoanthropologist, so I both do sort of theoretical evolutionary biology work and also field research in the Afar region of Ethiopia.
Thank you. And now, who has had the most significant impact on the field of paleoanthropology, and what aspects of the work do you find most influential?
Well, paleoanthropology, unlike a lot of other lab sciences, it's a science of discovery. So within paleoanthropology, I'd say the most influential people were the people that made the most important discoveries. So Raymond Dart in 1925 discovered Australopithecus africanus, and that put human evolution in Africa. So that may have been the single most important discovery in paleoanthropology. And then the Leakeys, who worked in Kenya and Tanzania, were extremely important, and they discovered many species over the decades. And then lastly, I'd say Don Johanson, who discovered Lucy. And Lucy, as a single specimen, may be the most important human fossil ever discovered. At the time, it was the oldest, but also because it's relatively complete, it gives us this amazing picture of things like locomotion and the arms and hands, the pelvis, which really we did not have any idea about before. So it established a new story, which is that we are, at least at that stage of evolution, sort of partially arboreal, partially terrestrial, and frankly, pretty small-brained. So it created a whole new idea about what early humans look like.
Thank you. And is there a perspective you hold that diverges from the commonly accepted view in biological anthropology?
Well, I don't know if it's different from other areas of paleoanthropology. It's definitely different than other areas of anthropology, in which I view humans as essentially animals who are, happen to have large brains but needn't. And over our evolutionary history, there were right now maybe 26 different species of humans. We radiated into all kinds of different niches, much as other animals have done. There's only one species left, but for most of our evolutionary history, we were doing pretty much exactly what other animals were doing. So I tend to take a fairly biological perspective on humanity overall.
You've contributed significantly to redefining the genus Homo, particularly through your work in Eastern Africa. So could you summarize what your research has revealed about the origins of our genus and how interpretations of Homo has shifted over the last few decades?
Sure. Well, we sort of knew that somewhere between 2 and 3 million years our genus appeared. And it's just one of these odd artifacts of preservation that there are very, very few places on Earth that preserve that time interval, especially the 2.5 to 3.0 million year gap. And it actually took us 10 years to find a spot that did preserve that well enough so that we had decent human fossils. And then when we found it, it turns out that Homo diverged pretty soon after the last appearance of Australopithecus afarensis in East Africa. In other words, it's a fairly ancient lineage. 2.8 million years is at least how old it is. Now, there is a debate in paleoanthropology, I suppose, about how we should treat these early Homo fossils. So some researchers have argued that they don't look enough like us to qualify for inclusion in our genus. In other words, they don't have particularly large brains. They weren't very sophisticated. They were small. They would have looked a lot like Australopithecus. But in evolutionary biology, generally, that's not how you determine which species are in different genera. Normally, you just say which animals follow which branch point. So in other words, every animal on this side, even if it looks something like the node, like the ancestral condition, still is in that group that follows from that split. So that's how taxonomy is done outside of paleoanthropology. Paleoanthropology has resisted that. I think that some researchers look at humans and see us as special or different, and therefore slightly different rules might apply. And as part of my general model that humans are animals, we should also be treating the taxonomy of humans the same way we treat any other group. And if that's the case, anything on the lineage towards us, Homo sapiens, like that diverge from Australopithecus, is Homo, and that means all of our early fossils are Homo, including fossils from Kenya and also our fossils from Ethiopia.
And things like the debate over whether Homo habilis and that type of material should be included in Homo, that fits under this umbrella.
Right, that is part of this debate. So yes, some researchers have argued that they shouldn't qualify, that there should be sort of a threshold, and you have to sort of pass a threshold. The problem is that any threshold is arbitrarily defined. I mean, you can say it's 600 cc's of brain. No, it's 700 cc's. No, it should be 800, right? So that's arbitrarily defined. Whereas if you use a branch point, it's not arbitrary, right? I mean, everything is on this branch or that branch. And, you just let the chips fall where they may. And, that's how we do it in every other area of evolutionary biology. So I think we should stick to that.
You've conducted fieldwork across other sites in both Eastern and Southern Africa. So could you walk us through some of the differences between collecting fossils in Southern Africa and Eastern Africa and some of the logistics involved in the process?
Sure. Yeah, I started working in South Africa, and down there, all the fossils are in caves. So, and frequently water has saturated the caves, and that water has calcium in it. So everything is sort of mashed together. They call it breccia. It's a combination of this hardened dirt and the fossils. And it's just very slow going. Sometimes the breccia is really hard. So it can be quite difficult to get the fossils out. The upside being they just have a ton of fossils. I mean, they really have a lot. In East Africa, it's all sedimentary deposits, which means if you can imagine your head sort of the edge of a lake, just mud settling in over the, thousands and millions of years. So when that erodes away, the fossils are just left there. They're not, packed in with anything. So when we look for fossils in East Africa, you just walk and walk and walk, looking down. And there are some areas that are fossiliferous, and you look among the non-human fossils and hope that there are human fossils sort of scattered in there. I think roughly, very roughly, it's 1,000 sort of animal fossils for each human fossil, because there just aren't that many large primates on the ground at any given time. It's just like chimps. I mean, there aren't that many chimps, in East Africa. So there probably weren't that many humans on the ground. So they preserved probably in the same proportion as other animals, but there were a lot more, grazing and browsing animals than there were humans. So logistically, oh yeah, sorry, go ahead.
No, I was just going to ask, and when you're going over and you think you've found a hominin, you can tell pretty quickly when you're looking down, or do you collect everything and then make a decision what to keep as you're going through?
Well, normally you identify what it is right away, and then we carry pin flags, and you put a pin flag down and you call the people around and they kind of do a surface crawl. You have to know your anatomy, obviously, and there are certainly some things that you find and you're not sure, and you collect it, take it back to the research tents and compare it to other, your comparative selection. Normally we bring comparative collections with us, so you can sort of figure out, but almost always you can tell what it is. Mostly teeth preserve, because teeth are harder than bone, and so they never decay. And we find a lot more teeth than almost anything else. And teeth for humans are very distinctive. You're never going to mistake a human tooth for anything else. So yeah, you know right away what it is. The main thing is just to see them. We work with AFAR, and they're kind of like the guards and fossil hunters, and people that work outdoors just seem to have better eyes for spotting things. They just, I sit in front of a computer all day and my brain sees things in sort of that two-dimensional space. But people who are outdoors all the time, they just seem to see things. And man, they find most of our fossils for sure. And we train them so that they understand the anatomy, and they call over and over, put a pin flag down, and then we collect what we can. Sometimes we screen, if we think there's something beneath the surface. So yeah, I mean it's I'd say a combination of everything. You will never turn down a human fossil if you find one. They're so rare.
Just moving on to a question about your research you've done on some of these hominin material. So your research also investigates the biomechanical and evolutionary constraints on the hominin, the craniofacial form. So could you discuss how recent advances in analytical techniques have enhanced our understanding of hominin cranial evolution?
Sure. Well, so one big area is biomechanics. So biomechanics is kind of the engineering structure of the body, and evolution selects for shapes because they're better at doing certain things. So that means if you understand the shape, you can infer backwards into what the selection was for. So, giant jaws and giant teeth, obviously the animals are eating something harder or tougher or more abrasive. And originally, back when I first was in grad school, everything was sort of modeled really simply using beams, and levers. And now we have things like finite element analysis, when you can sort of make a mathematical model of the entire skull, and you reduce the math to analyzing millions of little triangles on the skull. Each triangle sort of covers a small amount of space. And when you apply pressure to the whole skull, the deformation of each triangle gets a value. And so you can actually map the deformation or the strain across the skull itself using these sort of more sophisticated techniques. And that way you can get at where specifically on the skull selection acted to resist the pressures of hard foods, for example, which teeth, which part of the face, that sort of thing.
And for this, you need the well-preserved skulls from the fossil record to use, or is it particular morphologies that you need?
When we've done it, we use the whole skull. So yeah, you have to have at least one pretty well-preserved skull. Now, let's say it's 2/3 preserved or half. You can, mirror the other skull, kind of reverse it, and then, create a simulated full skull. And whenever this is done, no skull is really complete. I mean, they're always missing something, so you have to fill in. And so you'll take, if there's a chunk out of one corner, you'll take the opposite corner, mirror it, and then patch it in. And that's a lot of work. So for sure, when I was a postdoc, one of my jobs was exactly that, filling in all the holes in one of the skulls, Australopithecus skull, that was not completely enough preserved, so you didn't have to do it, right? So yeah, for sure. It's a lot of work. I mean, I spent, weeks and weeks and weeks just sitting there filling in tiny little triangles with the mouse, trying to make sure nothing was missing,
So the next round is quick fire questions. So all these questions are asked to all guests in the particular, so in this case, hominins. So which hominin species do you think Homo floresiensis and Homo naledi are most closely related to? So which fossil hominin or group that we know of do you think each are most related?
Yeah, floresiensis looks like it's most similar to the skulls from Georgia. So I think that's, you can scale those down fairly directly. And it's the idea is that once they got to the island, selection selected sort of downward on size, because there wasn't enough food to support a larger hominin. But it's what we have now. It's the best scale down. And you can imagine Homo erectus getting there. So I would say that's, like an early variant of Homo erectus, I would say. Homo erectus changed quite a bit over its entire time, like a really big skull later. But early Georgia, whatever, I think those are 800 cc skulls. So that's not that much of a decrease, compared to later when it's 1250 or something. Wow, naledi. That's a good question. That is a very enigmatic species. I would say habilis. It's somewhere in the habilis. It persisted, from the habilis early erectus period, and just didn't get, outcompeted somehow. And it was just a relic species by the time, we catch them at whatever it is in the hundreds of thousands of years. Modern humans were around then. So they're definitely not descended from modern humans. Yeah, I mean, and that's just based on brain size. I mean, they are Homo for sure, and they have very small skulls and they persist very late. But we do have places where relic species persist. I mean, the coelacanth, it's 500 million or whatever, 400 something million years old and still exists. In I guess this Indian Ocean.
And just a follow up on the Homo erectus. So you prefer to use Homo erectus for products. I know some people prefer to split it into different groups. How do you usually use Homo erectus in terms of when you're using it in taxonomy for different specimens?
Right. Well, at least like the traditional African and Asian, they're so similar. I don't think that you could separate them. I mean, they would have been separated by geography, but much like wolves in Asia and wolves in the Americas are the same species, just separated by geography. That's my read on that they were the same species. I don't think that anything would have about them that we've ever discovered would have caused them not to interbreed, in which case you'd say, yeah, they're just, same species.
And what's the most underrated fossil site or particular specimen in terms of its importance to understanding human evolution?
Underrated. Wow. Well, I mean, so Lucy is this really important fossil, right? But the species itself, I think, is underappreciated. Normally, when you are trying to understand like an ape species and you go into the jungle and you look at gorillas, you don't look at one gorilla, right? You look at the old, the young, males, females, all these different aspects of them, right? And Australopithecus afarensis is one of the few species where you can actually infer all that information because we have so many. We have big males, small males, females, multiple children, all different parts of the body. So it's one of the few species where you really get a snapshot of not just the anatomy, but, like the social group. You can infer the social group based on the size of the males to the females. I mean, that's like an amazing thing that you could infer sociality in an extinct organism, So, and we can tell how they developed based on the eruption of the teeth in the juveniles, all kinds of things. Like people don't realize how important that species is.
Thank you. And the next question, you just mentioned Homo erectus about the interbreeding between African and Eurasian samples. So the next one's something similar. So do you think Neanderthals and humans, modern humans, should be considered the same species, linking back to recent evidence of interbreeding?
I don't. If you look at the genetics of what we know anyway from like modern humans, like the Neanderthal DNA that we have in us, there's no Neanderthal Y chromosome and no Neanderthal mitochondria, which means that only a female child of a female modern human and a male Neanderthal actually survived. So that means, and it's safe to assume that all different, males and females would, I mean, they would copulate when they had the chance, but only 1 out of 4 survived. That means there's something in the genome that's causing separation. In other words, they are becoming genetically isolated and are in fact 3/4 isolated. So I would say, it depends on where you draw the threshold, but Neanderthals disappeared. There's no meaningful DNA in us, and only rarely could we interbreed. To me, that just says we're different species. Like I think the DNA answers that question.
Thank you. And the last question is always something outside your research directly. So you recently published a book called The Evolution of Everything. So could you share what inspired you to do this book and what are the central ideas you hope readers take away from it?
Right. So the basic premise of the book is that you can use science operating at different scales do not just explain sort of the physical world, geology and astronomy and biology, but also you can extend that into the present. So in other words, you can use scientific models to explain all aspects, a lot of aspects of human behavior. Those can be applied to things like war, for example. Like, and there are a lot of things, patriarchal social structures, all those things I think have biology underlying them. And one of the premises of the book is that our ability to overcome our evolutionary biases is what has given us the modern world. So in the Enlightenment, we overturned or started to overturn what had been the previous 10,000 years of, constant warfare, patriarchy, slavery. Those are all, I think, there are natural instincts driving those things, and our ability to overcome those things is fairly recent, just a few hundred years old. But that is what has given us the modern world. So it's kind of a call for people to recognize the biology, but also recognize that we don't necessarily want to be driven by our biology. And so how can we push back against that? So that's kind of a two-part thesis.
Thank you. And well, I'll ask the second question as well, because I've been asking this one to a lot of people that do hominin, so it'd be interesting to get your thoughts. So
Sure.
There's issues around access to fossil material and digital data, especially in hominin material. So how has this affected your own research, and what changes would you like to see in terms of data sharing and specimen accessibility across the field?
Well, right. In terms of digital data, the presence of it obviously means you can work from home. I mean, and you can have copies of things. And I mean, obviously that's just, the portability of information is huge. There has been a little bit of difficulty with specimen access, traditionally in paleoanthropology. I mean, if you spend, a decade finding a spot and you find a fossil, you should definitely have first right to describe it. And you should have a couple of years to do that. But at that point, it should become the property of, the property of the country and/or the property of the scientific community. In other words, the knowledge should be accessible to anybody. And that's what we do with our fossils. So we discovered it in 2013, published in 2015, and now anybody can look at it, right? But that's not always the case with fossils that people have discovered. And for sure, people have been sitting on fossils for a long time and preventing others from having access. Paleoanthropology is, because it has the potential, like any given discovery has the potential to give you, a modest amount of notoriety. It certainly attracts a certain type of personality. Now, I'm not saying everyone in paleoanthropology is this way, but there are certain people who, that's what it's about. And one way to leverage that is to prevent other people from examining your material, because you get to say, this is what I found. No one else can gainsay it. You're saying, oh, no, I'm doing further study. You can't look at it. It's not, it doesn't speak well of our discipline. There was a big article in The Guardian recently, I'm sure you probably read it, about Sahelanthropus that spoke to this to some degree. But it also happens in other places as well. It definitely happens in Ethiopia. And we try to work against it. But you do what you can. There are certain other places too where there are fossils and you simply can't access them. I've been trying to access the Georgian fossils for a while, and I can't even get people to answer my email.
That's what I was going to ask you. Like, when you were describing your material and trying to get, comparisons with other Homo specimens, was that a challenge just to get,
Well, I mean, we used the East African material just because it was the most relevant, sort of in terms of geography and time. But there's a bunch of stuff I would like to do with the Georgian fossils. It's just very difficult to access them. I regard every single measurement that someone takes of a tooth or a, brow ridge as its own scientific hypothesis that should be subject to check by other scientists. Because if you measure it incorrectly, then someone else should come along and get the right measurement. So I feel like we should all be able to access everything with our own tools and re-examine every single thing anyone's ever said. I mean, that's the foundation of science, right? Falsifiability. And, you know, it's funny, if you talk to people who work in other disciplines of evolutionary biology, they just can't believe us. I mean, they are slack-jawed at how, like, huge egos and unfriendly personalities, you know, shape it. Whereas, I don't know if you're studying, like, fossil turtles or something. They're all just like, glad to see each other. Hey, how's it going? Look at my new fossil. You know, do you want to measure it? Let's collaborate. It's just not like that in paleoanthropology, and it's a problem. You know, I mean, our particular field project, we have gone out of our way to not be that way, but that's not necessarily going to change the way others behave, especially people who are set in their ways.
Yeah, and I guess that's what that Sahelanthropus recent news article was covering, was how it's still going on even from samples from decades ago that was
Oh yeah,
found.
oh yeah. No, we just looked at for a paper. We have a paper coming out in, you know, maybe a month or something, and we're describing some dentition. And I was the first person to look at a particular specimen, and it had been discovered and described more than 25 years ago, and we were the first people to even ever get a chance to throw a caliper on it. And I, you know, I had to push a little bit to do it. So, you know, I happened to be in a position to do that because I know people in the museum and so forth, but I don't know anyone in Georgia. So, well, or only one person. And so, you know, and the woman who was in a position to help, she says, just be prepared. Like, it's going to go to the mat, as we say.
Thank you, Dr. Villmoare, for doing this interview. It's been a pleasure speaking with you.
Yeah, my pleasure. Take care.